Okay, so here we have a chemical with the name DACS3, which causes cell death in rapidly dividing cells like cancer. To investigate why certain cells can be killed by DACS3, scientists studied a cancer cell line called HAP and several different DACS3 resistant cell lines derived from the HAP cell line. Okay. The scientists exposed the cell lines to increasing concentrations of DACS3 and measured the average percent survival of each cancer cell line. So it seems to me that the first cell line to drop is the dashed line with the circles, which is the HAP. So the HAP is the HAP cell line is the most damaged by the treatment. Okay, very good. Which of the following best describes how enzymes regulate biological processes? So, do they make the activation energy higher? No, they make it lower, right? Do they increase? No, they don't increase it. Yeah, good. Okay. Based on figure 1, which of the following concentrations of DACS3 result in 10% survival of HAP cells? So a 10% survival, that would be here. Draw it to the dashed line with the circle in it, which is basically about 0.50. Okay, that's how much. Okay, question 39. B. Which of the following claims is best supported by the data in figure 1? Okay. In question 39, which of the following claims is best supported by the data in figure 1? So let's see figure 1. Let's just bring it here close to us. Okay. So at the DACS, as the D, is that at or as? I think it's at. At the DACS3 concentration of 1, okay, which is the x-axis being a 1. Okay, so here basically, forget that. Half as many HAP survive as the DACR. Let's look. At 1, the DACR, no, because the DACR are all at like almost 100%. But here it's like at 1, the whole HAP died. 0% lived, basically. So no, it's not true. As the concentration of the DACS3 increases from 0.05 to 0.10, the decrease in cell survival is greater for the HAP. That's actually true, right? They just dropped. So it's B. Based on the experiment, which of the following justifies the use of the HAP cells exposed to DACS3? Well, look, we already know what the HAP cells do, right? We already know that they're going to be cancer cell lines. So it's like your control, you know that the effect is going to happen on them, right? Yeah. So we're essentially comparing them with other cells, or we're comparing other cells to them. So, so the HAP will be used as essentially a control. Is that what they chose? What you chose? Okay. 41. The scientists screened the DACS3 resistant cells for mutation and found that they all shared a mutation in a protein called SDR2. So now the focus is going to be on SDR2. Normally, SDR2 catalyzes a reaction that converts each molecule of DACS3 into DAC cone. Okay, DA cone. DA coners cause protein misfolding and cell death. So like what I understand here is that to be able to resist the drug, you have to be mutated, right, in the SDR2 protein. Is it mutated? Right? And normally, so if the protein is not mutated, what the protein does is that converts a molecule of DAC S3 into DA cone. DA cone causes protein misfolding and causes cell death. That is only occurring if the SDR2 is not mutated, but right now it's mutated. So the mutant SDR2 is unable to catalyze this reaction, and that's why the cell is going to die. In a follow-up, is not going to die by the drug. In a follow-up experiment, the scientists exposed HAP cells to DACS3 and an allosteric inhibitor of SDR2. So if you inhibit SDR2, this substrate is no longer able to bind the DACS3. And you will no longer cause cell death. Okay? If you inhibit SDR2, you can no longer get DA cone and you can no longer get cell death. So you get less DA cone, right? You will get less, not more. So either A or C. Okay, so if you get less DA cone, so by getting less cell death, you will have more percentage of cells that are viable or basically alive. 42. Based on figure 1, which of the following statements best explains the change in the size of the population shown in region X? Okay, well, is death rate exceeding the birth rate at region X? Of course not. Relax. And are we starting to basically flatten out, let's say? Okay. Does the carrying capacity exceed? Does the size population exceed the carrying capacity? Here's the carrying capacity, right? So we're not even at it yet, right? Yeah. So it's not D. Okay, so it's not going to be... let's check. So basically, look, you should just put X here, which is like the midpoint. And anything after that, the rate is slowing down. And so, yeah. So, yeah. Because slowing down due to density dependent factors. Yeah, so anything after point X is going to be a slowdown. So it is going to be choice B. Okay, and density dependent factors means resources become limiting. Okay. Here we have pigeons. Okay, we got pigeons here. And if you were to mate the male with blue feathers with female with brown feathers, you get male and female to be all blue, meaning that blue is the dominant trait. But if you were to mate male with brown and female with blue, you get both colors. You get both colors, but males only blue, females only brown. So there is something about that which is going to be called Z-linked. Because how come we get exactly all the males to be blue, exactly all the females to be brown, right? So it's going to be Z-linked probably, or X-linked if it's a human. Okay. So males will have ZZ, females will have ZW. Okay. So we just established that it's going to be Z-linked, right? Because how can you just have exactly all blue males, exactly all brown females? So from the second cross. So it's going to be sex-linked, not autosomal. And we know it's dominant from the first cross, basically. That blue is dominant from the first cross. So yeah. So it's A. 44. Which of the following statements best explains why seeds swell when they're exposed to rainwater? So to swell, they have to take in the water, correct? Yeah. So this is the water, and this is the seed. So water has to go into them because water is hypotonic compared to them. Okay, water is hypotonic compared to them. So it's not going to be A, right? And it's going to be B because water always goes from the more water potential to the less water potential. Okay, 45. The protein TBK, I think that's 1, plays a role in regulation of both cell division and apoptosis, which is cell death. Increased levels of TBK1, so more TBK1 will give you cancer. Everything is cancer these days. You drink cola, you get cancer. You eat sugar, you get cancer. You drink water, you get cancer. I don't get it. Okay. Which of the following predicts the likely effect of an increase of TBK1 on cell division? So of course cancer means more cell division, correct? So it is B. 46. Which of the following best describes evidence that all eukaryotes share a common ancestor that's different from that of the prokaryote? Well, look for something that only eukaryotes have. So it's not D, right? Because eukaryotes and prokaryotes have ribosomes, and the same goes for the DNA. The same goes for the plasma membrane. So it is C. Okay. 47. The bearded lichen a symbiont association composed of fungi and algae, hangs from the branches of Douglas fir trees. Scientists hypothesize that the lichens require the surface of the trees to live, but neither harm nor benefit the tree. So the lichens don't harm or benefit the tree. That means the tree is experiencing commensalism, right, with the lichens, because the tree has zero benefit, but the lichens have a benefit. To test the hypothesis, they monitored the growth of Douglas fir trees before and after bearded lichens were removed. So look, if this is true, if commensalism is true, if you were to remove the bearded lichens, nothing will happen to the Douglas fir trees, correct? Which of the following supports their hypothesis that the lichens have no effect? So That means the tree growth before and the tree growth after is the same.
So it's P. I don't know.
So it should not change, right? It should continue like it is. It should just continue like it is. So it's B. Okay. Okay, so 48. Based on the model of sucrose transport into plant cells, which of the following is a testable question that would most directly help researchers determine whether a proton gradient is required for sucrose transport to occur? Okay, you want to test it, right? So which one of them can be tested? So look, if you want to know whether the proton gradient is important or not, all you gotta do is you have to block it. You gotta do what? You gotta block it. So A. Viruses infect host cells and use those host cells to make new viral copies. There's a family of viruses, commonly known as pox viruses, that contain a double-stranded DNA and infect only eukaryotes. Neither pox viruses nor their DNA can pass through the nuclear membrane. Pox viruses also lack DNA repair enzymes, resulting in a frequent mutation in their genomes. The rapid mutation rate of pox viruses allows the scientists to study the evolution of the virus as infections spread in and between natural populations of host animals. Based on figure 1, which of the following statements correctly describe the relationship within the family? Let's see. The sheeppox virus shares a more recent common ancestor with Mycosoma virus, whereas the sheeppox virus shares a more common ancestor with Mycosoma virus than with rabbit fibromavirus. Actually, no, because those two have the same common ancestor. So that means they will have the same date of the common ancestor divergence with the sheeppox virus. So it's not A. Swinepox virus shares a more recent common ancestor with the deep pox virus than it does with the goat pox virus. Is that true? Does the swinepox virus share a more recent ancestor with the deerpox virus? Well, look, so here they split, this one this way and this one this way. One went up toward the goat and one went down toward the swinepox. Swinepox shares a more recent common ancestor with deep pox than it does with the goat pox. So B is going to be wrong because swinepox shares the more recent common ancestor with deerpox. Let's see, swinepox and deerpox, they have to meet all the way here, versus swinepox with goatpox who meet here. How is this
Oh, yeah, that's right. C is right.
more recent?
C.
So it's not B, right?
It's C.
C. Let's look at C. Tanapox, right, which is this one, shares the same most common ancestor with deerpox, which is all the way here, as Tanapox shares with Mycosoma, which is also all the way here.
So it's C.
So it's choice C. 50. Which of the following best describes a measurement of the evolutionary fitness of a virus? It's not about how many proteins or diameter. It's about how many viruses you can actually produce. 51. Which of the following best explains why this virus family genomes must contain genes that encode enzymes required for DNA replication? They must have genes that encode enzymes, so the virus must bring its own genes that makes the enzyme required to replicate the DNA. So look, in the beginning of the question, we were told that pox viruses lack the DNA repair enzymes, and the pox viruses and their DNA both cannot pass through the nuclear membrane. They cannot enter the nucleus. That means they have to copy their DNA outside of the nucleus. And all the enzymes in the host required to copy the DNA are found inside the nucleus. So they better bring their own stuff with them. Yeah. 52. Viruses can gain different alleles of viral genes or new host genes when they infect and reproduce in host cells. Scientists are studying whether this process, known as transduction, has occurred within this family of viruses. Which of the following best explains why transduction could be beneficial to a virus? Well, it's able to gain different alleles of viral genomes. And of course, that would result in more diversity, right? And more diversity means more room for natural selection to work on something. So it is C. Okay. A student claims that the extinction of one host species that is typically infected by a member of the virus family may have no effect on the prevalence of the virus. You want to support the claim. Okay, so it has no prevalence on the virus. So that means it can have other hosts to look for, right? So if you kill one, if one dies, you also have a lot more to go and live in. Okay, here we have glucose becoming pyruvate, which then becomes ethanol. That's basically what we call alcohol fermentation. So, based on figure 1, which of the following best explains the relationship between glycolysis and fermentation? Well, fermentation is able to reproduce 2NAD plus to be reused by the cell. So you must be always cycling back NAD plus so it can be used by the cell. And the source of the NAD plus is the glucose. So it is C. Okay, so here it shows that fermentation temperature and ethanol, they affect each other. Ethanol produced during fermentation by certain microorganisms can be used as engine fuel. Researchers interested in maximizing ethanol production through fermentation investigated the effect of temperature on ethanol production in yeast. Based on the data in figure 1, which of the following best describes a follow-up experiment the researchers should perform to determine the amount of sugar that will result in the maximum amount of ethanol production? So that maybe there is more sugar, there is more ethanol produced, for example. Since we're looking for the maximum,
So it's just like,
yeah, you should just connect it with the maximum, which happens at 30 Celsius. So it's A. 56. Here we have lysosome working at acidic pH. We have cytosol working at neutral pH, almost. And as you can see that we have active hydrolytic enzyme inside the lysosome, but they're inactive outside the lysosome. That means that this enzyme is pH dependent and it only works in acidic pH. Which of the following explains an advantage of cellular compartmentalization? Do you know what this word means? It means locating your organelle with an internal membrane, within a membrane. So the inside does not leave the outside.
mitochondria is nice.
To the outside, yeah. In fact, anything with a membrane is called compartmentalized. So whatever is happening on the inside does not touch the outside. So having those internal membranes will be good to protect the rest of the cell from this acidic pH. Okay, so different environmental conditions. Cool. We have birds whose feathers color, which is genetically controlled, ranges from being dull brown to bright blue. The feather color distribution of population of male lazuli buntings was studied when the bird's habitat included many high-quality nesting sites, and many generations later, when their habitat had few high-quality nesting sites. Which of the following claims about the effect of the reduction in nesting sites is best supported by the data? So the feather brightness will be between dull brown and bright blue for the habitat with many high quality nesting sites. So it's going to be intermediate, right? Which means stabilizing selection. But if it's extreme, that's called disruptive because you have extreme on both sides. In the side of the dull brown, the side of the bright blue. Which of the following claims about the effect of the reduction in nesting site is best supported by the data? So basically, in the beginning, we only had many high quality nesting sites. So in the beginning, we only had the dotted line, but then over time, we shifted toward the extreme. And the extremes have dull brown and bright blue. So the extremes were the ones who actually got to mate or have more fitness, let's say.
So is it
Expend more energy maintaining their color. That's not.
yeah.
No, it's not D. And so yeah, like fitness toward the extremes, that means they have more fitness being dull brown and or bright blue. Chemical signaling between two nerve cells. Certain nerve cells release a chemical signal that diffuses through the extracellular fluid between two cells and binds to receptors on the plasma membrane of the second nerve cell, which then causes a response in the second nerve cell. The chemical signal can also re-enter the first cell through a transport protein. So it can re-enter through a transport protein, reducing the concentration of the signal in the extracellular fluid between the two cells. Which of the following drugs will likely result in an increase in response of the second nerve cell? So you have to block this transport protein for basically the molecule to stay in the synapse and possibly make it across to cell number 2. Yeah, so you must prevent it from entering the first cell. The proportion of each nucleotide observed in the coding strand of exon 1 and intron 1 of a gene is shown in table 1. Scientists use the data to compare the proportion of nucleotides containing G or C with proportion of nucleotides containing A or T. Based on data in table 1, which of the following is the ratio of the proportion of G and C containing nucleotides in exon 1 to the proportion in intron 1? So add G with C. In exon 1 and divide by G and C in intron 1. Right. So you're going to have 0.63 over 0.41. D. 60. Scientists investigate the mechanism by which a particular type of cell changed from its normal phenotype to that of a cancer cell. They found that the cancer cell contained a mutant CMYC protein that caused an increase in the activity of SIRT1 protein. And SIRT1 inhibits apoptosis and protects CYMC from intracellular degradation. So the SIRT1 protein blocks apoptosis, right? Which of the following predicts the most likely effect of introducing an inhibitor of SIRT1? So if you inhibit SIRT1, you will get apoptosis, correct? Would you agree? And that means you will have cell death. So it's not D. And it's not B. Will you affect the levels of CMYC? That's the question. So SIRT1 not only inhibits apoptosis, it also protects CMYC from normal degradation. So now by blocking SIRT1, SIRT1 protein, you will not only cause cell death, but you will also you can no longer protect CMYC from normal degradation. Okay?
So it's C.
So it is C. Because now they will start to die.